Frederick Griffith conducted an experiment involving two strains of Streptococcus pneumoniae bacteria. The first was the smooth S strain, which contained a polysaccharide coating that protected the bacteria from the mouse's immune system, making them virulent. The second was the rough R strain that lacked a polysaccharide coating, which were not protected from the mouse's immune system and were therefore non-virulent. In one part of this experiment, Griffith injected lab mice with dead S bacteria that had been killed prior to injection using heat. The results of this part of the experiment had shown that the mice that received the injection survived and were not harmed. In a later experiment, Griffith created a mixture of heat-killed virulent S strain bacteria and a live non-virulent R strain bacteria. Surprisingly, after injecting the mice with this mixture, all of the mice had died. After observing this, Griffith concluded that the non-virulent rough strain bacteria in the mixture had to absorb what was called the transforming principle from the dead virulent S strain bacteria, which changed the non-virulent bacteria into virulent bacteria. At this point in time, Griffith did not know exactly what this transforming principle was. In 1944, Oswald Avery, Colin MacLeod, and McCarty set out to identify the chemical nature of Griffith's transforming principle. At this point in time, the three contenders for the transforming principle proposed by the scientific community were proteins, RNA, and DNA. Most scientists believed that the proteins were the transforming principle, considering the vast amount of different proteins in existence as well as the fact that they had specialized shapes and functions. In addition, proteins constitute a significant percentage of every cell's composition, measuring in at levels of 50% or above, whereas RNA and DNA constitute levels of around 20% and 3% respectively. To begin their experiment, Avery, MacLeod, and McCarty used heat to kill virulent S strain bacteria, similar to Griffith. The bacteria were then homogenized and filtered to ensure uniformity. Three different enzymes that break down proteins, RNA and DNA respectively, were added to three different samples of dead virulent S strain bacteria, one enzyme in each sample. The enzyme RNase destroys RNA, protease destroys proteins, and DNase destroys DNA. Each of the samples were then added to individual cultures of non-virulent R strain bacteria. The scientists knew that if after the R strain bacteria were introduced to the sample of treated dead S strain bacteria and they still transformed, then the material that the specific enzyme destroyed in that sample could not be the transforming principle. The samples containing RNase and protease caused the non-virulent R strain bacteria to transform upon introduction. The sample containing DNase did not cause the non-virulent bacteria to transform, meaning that the transforming principle had been destroyed upon the addition of DNase. Avery, MacLeod, and McCarty concluded that DNA is the transforming principle. They knew this because upon the addition of DNase, the non-virulent bacteria remained non-virulent, as they were not transformed following the destruction of DNA in the heat-killed R strain bacteria.